Today's topic is going to be on DNA fingerprinting. DNA fingerprinting is what we are going to discuss today. DNA fingerprinting is one of the greatest identification systems we have to recognize an individual or living organism. Every living creature is genetically different from others in its own way, except for identical twins or identical triplets. DNA is comparable to a serial number for living things because there's no two people in the whole world that have the same fingerprint, that have the same numbers or the same code, the same fingerprint code that another person has. So, and because of this, each individual contains a unique sequence that is specific to that person alone. And this one thus increasing its effective use in forensic and the probability of finding an exact match. DNA, also known as deoxyribonucleic acid, contains a specific sequence of bases called nucleotides from the word nucleus, which contain the information of all the characteristics of living organisms. This information was inherited through the DNA of their parents, that is, from the father and from the mother. DNA is found in almost every cell of living organism. In human being, it can be found in the blood, it can be found in the tissue, can be found in the hair, or any part of the body, the bones. Even the saliva, the semen, all this one contains the DNA. So, the DNA represents the instruction book for making living organisms. For example, this DNA contains instruction that will give instruction to the organism, for example, to human being, on whether to grow tall or to grow short, whether to be black or to be white, whether for the hair to be blonde or for the hair to be negroid hair, and so on and so forth. Whatever you see in a man is because of the information inside the DNA. It is because of the DNA instruction. So, the four nucleotides that we mentioned that constitute the sequence of DNA are: number one, adenine, represented by A. Let me expand this one. You have adenine here, represented by A, and this adenine bond itself exclusively with thymine. Look at thymine here. Thymine is denoted by T. And also guanine, which also binds itself to cytosine. Guanine is denoted by G, and cytosine is denoted by C. So the molecular structure of DNA can be imagined as a zipper. I said before we can understand fingerprint, we have to understand the DNA basics. And we said that the DNA contains 4 nucleotides that constitute the sequence of DNA. And these nucleotides, they are adenine, denoted by A, which bonds itself exclusively with thymine, and thymine is denoted by T. And also we have a guanine that we denote by G, which also binds itself exclusively with cytosine. Cytosine is denoted by C. So when you see this, you see that adenine and thymine, they are joined together. That's how this thing, one way or the other, they twisted with one another. The nucleotide. And this molecular structure can be seen as a zipper. As a zipper. Look at the board. Let me try to expand it so you can, put it right so I can see it. So if you look at your screen, you have a figure here. The figure here is zipper. Is zipper. So the molecular structure can be likened to a zipper that has the teeth, and these teeth enter into one another. Enter into one another. So we have adenine, we have thymine, we have cytosine, and then we have guanine as the nucleotides. So this sequence form the molecular structure. It forms the molecular structure, and the structure can be likened to a zipper. To a zipper. Look at this figure. Just imagine the sequence is like a zipper, with each tooth representing one of the 4 letters that we mentioned. The adenine represented by C, the cytosine represented by C, the guanine represented by G, and the thymine represented by T. So all of them A, C, G, or T. So with opposite teeth forming either of the 2 parts, that is AT. Look at it. AT, CG, GC, TA, like that. That is how the sequence moves. Now, a chromosome. Perhaps you might have heard of chromosome before during your biology class in maybe in secondary school. You have a chromosome is common because it's part of what we studied then in secondary school. In biology, in cells, it's part of cells. A chromosome is the physical state of genetic material during the division phase of a cell. Humans have 23 pairs of chromosomes. The same way we were taught then in biology, which makes 46 individual chromosomes. That is 23 pairs. That is 46 in every human being. Half of this, that is 23 pairs, is from the father, and the half of it, which is also 23, is also from the mother. So chromosomes are found in the nucleus and contain a linear strand of DNA. The DNA molecule is twisted onto itself, as we see it here, twisted onto itself, and the supercoil molecule is enclosed in protein which help maintain its shape. The chromosome carry the genes that make each individual. Now, that is the basic. Now let's go to now that we have a better understanding of what DNA actually means, let's move on to the basic of making a DNA fingerprinting. A DNA fingerprinting. There are 3 types of DNA fingerprints. 3 types of DNA fingerprints. The first one is called RFLP. RFLP. Look at it in your screen. RFLP. And the second one is VNTRS. And the third one is STR. So there are 3 of them. FRLPS, FRLP, VNTR, and STR. Now, let's move ahead. Restriction fragment length polymorphism, or RFLP as they are commonly known, were the first type of DNA fingerprinting which came into the scene in the mid 1980s. So RFLPs focus on the size difference of certain genetic location. The first step in creating an RFLP is a fingerprint is obtaining and isolating the DNA. I repeat, the first step in creating an RFLP fingerprint is obtaining and isolating the DNA. DNA can be then obtained from almost any part of the of human body, maybe any part of human body, the cells from the nerve, from the skin, from the brain, from the hair, and so on and so forth. So now, after obtaining it, the first thing to do, before I go to that one, I want to tell us that we don't need large quantity of the human, maybe blood or flesh to get DNA from. All you need is just small quantity, a small quantity of the cells. So after obtaining it from human, maybe the cell of the blood, tissue, or any part of the body, the first thing to do is to extract from the tissue or the blood sample of this DNA. So after extracting the DNA from the sample, then we go to the second stage. And the second stage is the cutting. I mean, it involves the cutting, the sizing, and sorting of the DNA sample. I said the first stage, after obtaining the sample that we need, is extracting. The first stage is extracting the DNA from the sample. And I said the sample may come from the hair, it may come from the blood, it may come from any of the biological any of the part of the body of human body. So after obtaining it, you extract the DNA from it. That's the first. Then the second step or the second stage is cutting, sizing, and sorting of the DNA sample. The DNA is cut using restriction enzymes, which cuts the DNA strand at specific places. Restriction enzymes are usually isolated from bacteria that use them to degrade foreign DNA like viral DNA. Each type of restriction enzyme recognizes and cut a particular DNA sequence. So the DNA at this point is cut into various array of pieces, or various arrays of just it you cut into small pieces like that, and then after that one it is sorted into sizes through a process called electroprocesses. So after the thing might have been extracted, then it will go to the second stage, which involves cutting, slicing, and sorted. And the process is done through what we call electrophoresis. Now, in this process, the DNA particles are mixed into a buffer solution, and applied to a gel made from sewage agarose plant. So each side of this gel is connected to an electrical current. The DNA is negatively charged due to its phosphate groups. So it migrates towards the positive electrode or anode. The smaller pieces of DNA move faster, or let's use a common language. It will sift it through the gel than the larger ones. So this provides the basis for the fragment separation. This technique is the DNA equivalent of screening sand through a progressive finer mesh screen to determine particle sizes, as if you are using a mesh to screen particles of sands that will make some bigger one to stay behind and the smaller one to go through. So it's just like that. So the band, there's going to be a kind of a band pattern. So the band pattern that the DNA creates in the agarose gel is then transferred to a form of a sheet, a nylon sheet. A nylon sheet. To complete this transfer, a nylon sheet is placed on the gel and left to soak over the night in a high salt solution. Just leave it to soak up over the night. Then after that one, after the soaking procedure, the nylon membrane contains that contains the same pattern of DNA is as occurring the original gel. What really happens is this: the nylon sheet, we have the same pattern that is on the gel. The same pattern of DNA sequence will be transferred to the nylon, because it has been with it over the night, be on it over the night. So it will transfer the pattern on the nylon. So what you have on the gel, on the original gel, is what we are going to have on the membrane, the nylon. So after that one, the membrane is now prepared to undergo its probing phase. The probing phase is the purpose of the probing phase is to bring out, to compare what you have there, that pattern, with the pattern of the exemplar, so that both of them, you can see that both of them are the same. So we use radioactive or fluorescent label probes that are already hybridized onto the nylon membrane, which bind to specific DNA sequences present in the pattern to produce a pattern of bands, which creates the DNA fingerprint. This process can be performed with several pattern probes, and what we are going to have is what is likened to what you have here on your left hand side of the screen, for RFLP. RFLPs, that is what we are going to have there. So the pattern will be like this. This is exactly a fingerprint that has been completed, fingerprinting that has been completed. So it's just like it looks like a code, a barcode that you have in retail stores on products, and this is meant for is from a cell. It's a DNA from a cell of an organism, and you cannot have the same from another cell from another organism. So the code is unique. So it's like every human being has a serial number that make that organism unique from all other organisms. So the same thing with every human being. Every human being has its own DNA that is like a serial code that differentiates or distinguishes him from other human beings. Then there's another method that can use for fingerprinting, which is VNTR. VNTR. VNTR, variable number tandem repeats. Variable number tandem repeats, or VNTRS, represent specific locations on a chromosome in which tandem repeat of 9 to 80 or more basic repeats a different number of times between individuals. These regions of DNA are readily analyzed using the RFLP approach, the same approach that we use in RFLP is what we are going to use. The same approach is what we are going to use in VNTRS, VNTR. So use the same approach. And a probe to a VNTR locus. The fragments are a little shorter than RFLP piece. So what am I saying in essence is this? The same method is what we are going to use for the two of them, the same approach. But what really happens is this: in variable number tandem repeats is just to locate some specific places in the sequence, some specific locations in the sequence that repeat itself in a sort of pattern. That is what we are going to. That's for why we have what we have here at the right hand side. Look at the figure here. So if you use this, do the same approach, but what we are looking for are not the same. So we use the same approach and we get this result. As many times as we repeat it, we are going to get the same result. So since RFLPs and VNTRS are created in the same version, they exhibit the same overall advantages and disadvantages. That is, some of the advantages of this type of DNA fingerprinting are that they are the most stable and reproducible. That is, they are stable and reproducible. That is an advantage for this. All of the RFLPs and VNTRS arrows, they are reproducible and they are all also stable. This is a valuable trait to have when you are trying to determine an exact match of a person's DNA, which must exclude billions of other people with a certain degree of confidence. Another advantage is that they are also easier to prevent from contamination, since the DNA sample is larger. So the DNA sample that we are going to extract will be larger. So the advantage of that is that it can prevent contamination from affecting the result. So let's go ahead. Let's go to the third one, which is STRS. STRS. That's the third one. Recall that we said we have three methods of fingerprinting. The first one is RFLAP, and the second one is VNTR, and the third one is STR. Currently, the most popular method of DNA fingerprinting are short tandem repeats that we shorten to STR. Unlike VNTR, which analyze mini satellites that have repeat sequence of 9 to 80 base pairs, STRs, they use microsatellites which have repeat sequence of only 2 to 5 base pairs, introducing the less is more philosophy. The less is more philosophy to the world of DNA printing, fingerprinting. That is, even though you may have a little quantity, yet you are going to get good results. And apart from that one, because it is easy to use another process that you call PCR to use it, it is easy to amplify the DNA. The little DNA that you get from using STR, you can amplify that, you can replicate it. That is making like a copy of the DNA, which means when you get to a crime scene and you see just a little quantity of the DNA, it is possible for you to get a good result. But in the other two methods, you must have at least a tangible quantity. So now we are able to analyze a very small sample of DNA that is quicker and easier than any previously known method and match it to a person's identity. PCR was developed. That is a method that you can use to amplify it, to amplify the DNA sample. PCR was developed. PCR means polymerase chain reaction. So the PCR was developed in the mid-1980s also, and use the same principle that cells use to replicate DNA to amplify the specified region, which is usually between 150 to 3,000 base pairs in length. In order to amplify the DNA, to amplify the DNA sequence, a pair of short primary sequences, which are complementary to the ends of the targeted sequence, a special heat-resistant DNA polymerase called Taq polymerase. And the solution of the 4 DNA bases are all mixed together in a tube, or that is test tube, which contains a few copies of targeted DNA sequence. Then the DNA is amplified or replicated or copied or duplicated by the repetition of a cycle, which contains 3 vital steps. And the steps are: the solution is reheated to 95% degree centigrade to unseal the double helix DNA structure. This is what we know by helix DNA structure. This structure that you have here, this is double helix DNA structure that you are referring to. So to unzip it, we heat it to 95 degrees centigrade. Then we also allow it to cool to 55 degrees centigrade to allow the primers to bind to the ends of the DNA. The primer that were added to it will be allowed to bind to the ends of the DNA. Then Later, the solution is then reheated to 35 degrees centigrade, which is the optimal temperature for the tag polymerase to create a new copies of each DNA strand. So as depicted by this heated to 95, from that the DNA strand will separate. After that one, we are allowed to cool. It will cool down to 55 degrees centigrade. So as for the primer that was added to it to bind to the DNA strand, then later it will be heated again. And there you have copies. Look at it. You have copies. This is 2. Those 2 DNA strands have been duplicated into 4. The 4 also duplicated into 8. So and so on and so forth. You can repeat as you are repeating the cycle. You have more of DNA strands. The procedure avoids the lengthy probe hybridization that was used in the previous approaches. That is that of RFLP and VNTR. Since the whole PCR process takes only a few hours, that is the process of replicating it just take like 2 hours to complete, compared to RFLP or VNTR probe. So compared to that one that will take that will take a whole day, and the whole process in that approach will take like days. This one, within some hours, you can get a result of STR. You can get result of fingerprinting from STR. So this method, this STR is faster, and it is also easier. And the only disadvantage in it is that it is sensitive to contamination, because it is so small, so it is easy for it to be contaminated. And that is why at times, if you suspect that there is contamination in the process when you are using STR, then you need to use you amplify it, then use either of the 2 previous methods, because those 2 methods, they are not affected by contamination, but this STR can be affected by contamination. So but you have to do something to prevent contamination, and that will be discussed later in this lecture. The next thing is to talk about application of DNA fingerprinting. DNA fingerprinting is used in a variety of applications all over the world. They can be used to solve criminal cases, which is the primary thing that concerns us. In cases such as rape, it can be used to also used to conduct a paternity test, or even to use to determine the authenticity of a rare sport memorabilia. But that one does not concern us. Let's just limit ourselves to to crime scene investigation, or to criminal investigation. Whatever the case, it is evident that DNA fingerprinting has revolutionized the way the world identifies biological matches. We will discuss few of this one in the course of this study. One of the first accepted uses of DNA fingerprinting was in the investigation of sexual assault and rape cases. Detectives only had to match the DNA of the semen found at the crime scene with the DNA of any potential suspect to determine who was guilty of committing the crime. So, a DNA sample from the rapist could be obtained from any part of his, but for the victim can be, if it is from the victim, it can be obtained from the vaginal swab from the victim or any other semen that was released in the area. It may be from the semen may be released on the clothes or any of the clothing of the victim, or even on the carpet. So it can be obtained from that. But at times the best thing is to collect the underwear used by the victim immediately after the rape case. That one also can work. Now look at what you have on the screen. This is an example of an investigation, fingerprinting of a case of a rape case. In the figure we are looking at here, you have two suspects, so suspect A and B. So we take we have suspect A, we have suspect B on a rape case, and the clothing from which you have the semen also was used. We extracted the DNA from it, and the vaginal swab from the victim also, and also we took the sample of the blood sample of the DNA from the blood sample of the victim. Then we have a control sample. The control sample is a cloth that was worn by her. Also, we took, whether you can see DNA from it. Then, so we work on all these samples, and at the end of everything, this is what we are able to get from suspect A. Now, this is a look at it. This is from the vaginal swab. We have this. This is pattern that we have. Now, from the suspect A, we have this. So this one didn't match this one, so it couldn't have been him. Then the suspect B, from suspect B, this is what we have. Can you see that it matches this one? Look at it, 4 and 6. Then the cloth, the semen from the cloth, the same pattern. Look at it. So this is the blood of the victim. So it's not so what you have here. What you have here is from the swab from the vagina, from the suspect, and the clothes that you collected from this victim. All of them are similar. The fingerprinting from each one of them are similar, which means the deposit onto this victim, on the private part of this victim, is from the suspect. So that is a kind of result from fingerprinting. So that's a way of applying the knowledge of fingerprinting to solve a rape case. Let's mention maybe one again before we go into the next thing. We talk about DNA paternity test. Okay, let's leave that one because of time. Let's go to this one. Forensic science is the art of piecing together a crime scene in order to determine how the crime was committed, who was responsible, and at times how the thing was done. So when we have suspects, when at a crime scene like this, you have suspect A, suspect 1, suspect 2, suspect 3, and suspect 4. We have about 4 suspects, and DNA of each one of them were taken. What you have on the crime scene is this. This is what you have in the crime scene. And each of these suspects, their DNA were extracted and processed and fingerprinted. And now, after fingerprinted their DNA. These are the results. For the first suspect, this is what you have. It doesn't match this one from the crime scene. From suspect 2, this is what you have. It doesn't match this one from the crime scene. Look at from suspect 3. You can see that they are the same. And suspect 4, of course, is not the same. So from this result, you can see that the suspect in the suspect 3 and what you have, what was left in the crime scene, this the evidence that you have in the that you looted from the crime scene, and shows that suspect 3 is the source of this evidence. Is the source of this evidence with the DNA that you lifted from the crime scene. So that is the method. So let's move ahead. On the methods of collection of forensic DNA and collection methods. On this collection method, we are not going to do practical today because we have done the practical in about 3 weeks back in a topic that says discuss about the crime scene method. Yes, in the crime scene method. So if you go back to the crime scene method, you will see it there. Now let's talk about some things that may help us especially when we are using the last method, which is STR. So how do we prevent contamination? In fact, contamination is one of the greatest risks that the evidence must be guided from. If your sample of evidence is found to be contaminated, it will be thrown out of the court, as evidence that is contaminated cannot be accepted in the courtroom. So contamination can occur at the crime scene during packaging. You know, the last time that when we were discussing about when we were demonstrating how to collect sample from the crime scene, I told us that we should be careful while packaging, that we should package where we should use PPE equipment to protect our DNA from being transferred to it, and at the same time, when we are each time we are packaging, each time we touch anything from the crime scene, different items from the crime scene, we must change our glove so that we not transfer DNA from one object to another. So if you have 2 or 3 objects from the crime scene, we have to change our gloves. We change gloves. We change gloves so as to protect ourselves from contaminating the evidence. Then, apart from that one, when you are leaving the crime scene and you still need to go to another crime scene, everything that you use that are not disposable, everything that are reusable in that crime scene must be decontaminated. Things like even fingerprint brush must be decontaminated. Your suit, the jacket that you use must be decontaminated. The shoe that you put on, everything that you use, just everything that you can see go and be used elsewhere must be decontaminated. Even your notes, everything must be decontaminated. As we said in the other class, that one of the potent method of decontamination is to use jig, that is bleach. It's a potent method. So wash those things in bleach, or just immerse them in bleach. The contaminant will go. Now, at the crime scene, one of the factors that must be considered in trying to prevent contamination is the mother nature. At times there may be, if the cancer is outside, rainfall may dilute the DNA sample. May dilute the blood sample that is found on the cancer, or the biological fluid that is found there, and that may make it difficult for us to see a quantity of DNA that will be usable. So another thing that can happen if it is outside is that a piece of evidence that may contain DNA, like the hair or a piece of clothes that have been touched by the suspect, might have been blown away by wind. So another thing you must take note is the issue of preventing human factors. People that may want to come, relatives that may want to come to that scene, to that crime scene, supposing if maybe it's a murder case, some relatives, some neighbors may want to come and sympathize. This sympathizer may come and take the evidence. So in order for that not to happen, the first thing is to barricade the crime scene. Barricade it with crime scene tape, and at the same time let there be instruction that nobody should enter into the crime scene in order to prevent contamination. So that said, in the storage method of DNA, because DNA is a biological material that can degrade under certain conditions. DNA is so sensitive to its environment. So when you are transporting it, it must be transported is in a paper bag evidence bag, paper evidence bag, an evidence bag that is made up of paper, not nylon. And before even packaging it, it must be air dry, not sun dry, and not heat dry. You just dry it with air dry it, and package it inside an evidence bag that is made up of any material that is not nylon, because nylon or any material that can bring heat will degrade it, and if it is degraded, it may not be usable at all. And at times, DNA at an age crime scene may be difficult to process, to recover. It may be difficult to recover. So what you do is this: in a case where the crime scene, where maybe it's a homicide case or murder case, and the cadaver there is already decomposing, all we need to do, because at that time you cannot use the blood again, the blood might have degraded to a level that it will not be usable again, and even some of the flesh. So what we need to do in that case is to get the hair on the head of the dead body. The hair will be pulled off. We still have some flesh at the base of the hair. Then another thing you can use is the teeth of the dead body, or any of the bone will contain a large amount of DNA that will be used. So that's how you take it from, you take the DNA, we obtain the DNA from the hair or from the bone, either the teeth or the bone of the cadaver. So we are going to stop here today, and the next class is on is another topic entirely on case analysis.